All right, so we're going to talk about enzymes in this video. So enzymes are really just, they're proteins. And so you can see in this picture here, this blue whole thing is this enzyme, this big three-dimensional globular protein. And they're also, what their job is they act as catalyst. And they do that by lowering the activation energy, which we'll talk about in a second when we look at the graph. Energy of the reaction. And so what they do is they actually have this substrate or substance that they're going to try and either break apart or put together. So we'll look at the two different options. And that substrate binds into its active site, and it's called the induced fit model. So then you can see how like this enzyme kind of envelops over that substrate, and then it can break apart. And so this is what we call the enzyme substrate complex, when those two things are bound. And then eventually the substrate's actually going to be released. But the whole thing is that they're just trying to lower the activation energy of their reaction. They're used all over the place in this cell. And generally their names end with ASE. So like sucrase isn't the enzyme that breaks down sucrose. So you can see. So let's look at an energy curve here, a reaction curve. So you can see over here is we have our enzyme and our substrate. And this is the normal non-catalytic reaction. So we have to put in a lot of energy in order to get our product over here. But with our enzyme, it lowers the activation energy. It lowers the hill that this reaction has to get over. So there's less energy input needed in order to reach our final product down here, that it has a lower energy state. So the whole thing is it just makes that hill easier to climb. This enzyme does that. And the other thing you're going to notice is this enzyme, look, we've created a new product, but this enzyme is not used in the reaction. So they're lowering the activation energy. So we can say that again. Lower the activation energy. And not used up in the reaction, which is good because we can use them over and over and over again. This enzyme can now go grab a new substrate and do this whole reaction again. So let's look at the different types of reactions that can occur. So this is where we're going to take a substrate, and that substrate binds to the active site. We have our enzyme substrate complex for a brief moment, and then we create two products. So in this case, this is a breakdown, right? We're breaking something down. Enzymes can also put things together. So you can have two substrates and they bond to their active site. And then we have that enzyme substrate complex and we've created a new product. So we're creating something. Either way though, what they're doing is they're putting that induced fit model is that enzyme is putting some kind of stress on this bond here. And so it's allowing it to break apart. Okay. Or in this case, it's putting some these molecules so close together that they have nothing else to do but create this product. So it lowers that activation energy, whether you're breaking something down or you're creating it. So let's look at this graph here. So you can see there's a couple different things on this graph. So there's the S stands for substrate. So we're looking at concentrations of things. So the substrate concentration at the beginning of the reaction is really, really high, right? Lots of substrate in our system. The enzyme concentration is somewhat high, and then it dips a little bit. We'll talk about why in a second. And then it returns to that exact same level because that enzyme is not used up in the reaction. So E is for enzyme. And as we don't have any products at the beginning of the reaction, but as time goes on, we create a lot of products. So P is our product. And let's talk about that weird little dip. Why did we get this little dip? Because really, those enzymes are actually still there, but what they've done is they've bonded with our substrate and formed our enzyme substrate complex. So that's where those enzymes are. So where are they right here? Why do we dip? They're actually still there. They're just bonded to the substrate. So if you combine these two, it would create all those enzymes. You'd get the same number. So that's the enzyme substrate complex. So as time goes on and you make more and more products, you have less enzyme substrate complexes, even though you have plenty of enzymes because you have less substrate. Now, you want to be able to control a reaction as a cell. So there's ways you can control it. So one way is you have what's called competitive inhibition. So you have an enzyme, and then you have a substrate that looks like this, and you have an inhibitor. This is competing for a spot on the active site. So it binds to the active site. So it competes for spot on active site. Okay? It binds to the active site, the competitor, and it prevents the reaction from happening. See, no reaction. It directly blocks the active site. A non-competitive inhibitor binds somewhere else. So it actually binds to a different spot. It's called the allosteric site. So this spot here, the site on the enzyme, I'm actually going to draw it over here where you can see it. So this spot here is called the allosteric site. So it binds somewhere else. Somewhere else and changes the shape of the enzyme. Either way, it's preventing the substrate from binding to the enzyme and creating a reaction. The last part of this whole enzyme thing is there's this thing called allosteric inhibition. We just saw that. That's actually really what we just saw is allosteric inhibition. That's another name for that non-competitive. So allosteric inhibition is, it's the same as non-competitive. But you can see there's my active site, there's my allosteric site. My allosteric inhibitor binds to that. It makes my enzyme change its shape so the substrate can no longer bind, no reaction can occur. Okay, so there's this no reactions happening, no reaction. Versus this one where, here's my allosteric site. This is allosteric activation. So when the active site can't fit my substrate, right? This little funky little business cannot go in there because enzymes have that's the last part I should have mentioned at the very beginning, is enzymes are specific. So they have a very specific shape that is specific to that substrate. Just like any other protein, they're very specific. So sucrase can only break down sucrose. It can't break down lactose or glucose or any other, it can only break down sucrose. So very specific shape, very specific. So then this allosteric activator comes and it creates the active site, the correct active site, the non-distorted. So then the substrate can bind, and then now my enzyme, so we can get a reaction. So an activator helps a reaction occur, and an inhibitor prevents a reaction from occurring. Pretty simple and straightforward. So enzymes are super important in your body. They pretty much do everything in your cells that you need them to do because they lower that activation energy and kind of help that process. So that's it.